Hi, my name is Alexa Soares. I'm a fifth-year behavioral neuroscience major, and I'm completing my honors thesis in the developmental neuropsychobiology lab under Dr. Heather Brenhouse. In our lab, we research the effects of early life stress. This is an important area of study because almost 40% of children experience childhood adversity, and this has been linked to many forms of mental illness. We are working to understand how early life stress affects development and behavior later in life, in the hopes that treatments can be created to help people who have suffered from childhood adversity. In our lab, we model early life stress in rats subject to maternal separation, which simulates childhood neglect. With this model, I have found that early life adversity increases oxidative stress in the prefrontal cortex, the part of the brain responsible for self-control and decision-making. You can imagine how damage to this brain region could lead to many forms of psychological dysfunction. My data suggests that antioxidants could be used to treat people who have suffered early life stress and potentially prevent the emergence of mental illness. Through my research, I have found a potential mechanism driving the development of neurological dysfunction in rats that have experienced early life stress. Understanding this mechanism is the first step in creating treatments that can help people who have experienced childhood adversity. My research lays the foundation for a new approach to tackling this problem. Since early life adversity is linked to oxidative stress, it's possible that antioxidant treatment could be provided to people who have had adverse childhood experiences in order to prevent the development of mental illness. Currently, there are no effective biological treatments for preventing mental illness in people who have experienced early life adversity. There are medications available once a person has been diagnosed with the disease, but besides talk therapy, nothing can be done before symptoms emerge. Furthermore, many are hesitant to provide brain-modulating drugs to children. By focusing on oxidative stress, my research opens the potential for antioxidant treatment, which could be as simple as a modified diet rather than psychiatric medication. This is a new and exciting avenue of treatment for those who have suffered early life stress. I began this project over the summer, and I have come a long way since then. I had to spend a few months troubleshooting my protocol, and I hit another obstacle when our microscope broke in the middle of my project. Eventually, it was fixed, and I was able to complete data collection and analysis on all my groups. I have already presented my research at the Neuron Conference in Quinnipiac, where I won an Outstanding Poster Award. I'm looking forward to sharing my work once again at RISE. So far, my research has been focused on the prefrontal cortex, but I want to expand and look at other areas of the brain implicated in mental illness, such as the hippocampus, which is involved in memory. I'd also like to investigate antioxidant mechanisms and potentially test out an antioxidant treatment on rats that have experienced early life stress. I plan to write up my findings and publish them in a peer-reviewed neuroscientific journal. I also intend to continue disseminating my research through conferences like RISE and the Society for Neuroscience meeting in the fall, because I believe that communication and collaboration between scientists are essential to furthering progress.